Great. Well, we have time for several questions this morning. The only question you cannot ask is Sai Kumar directly when your sequencing run will be finished, because I think he gets caught.
And for Kevin, maybe this is too personal, but I'd be really interested in learning how, as a bioinformatician, I could actually get the NVIDIA GPU cores working on, say, a sequence alignment to the genome kind of a problem, or the kind of things that I do a lot that it seems like there's resources there that I haven't learned to tap. And do you have like workshops maybe that people here could learn from?
Actually, at some point we'd like to set up workshops on the NVIDIA question. NVIDIA has been developing porting various bioinformatics tools onto those platforms. The current NVIDIA platforms that we have are K20. We haven't seen too many people utilize them yet. And the key issue there, I think, a lot of times is if you look at the memory on board these accelerator cards, they're pretty small. And so if you're trying to process a large amount of data, or your genome sequence or whatever you're trying to do is larger than what will fit in memory there, you're better off using a standard CPU-based system. So, but there are some cases where it's probably going to be beneficial.
And it's hard.
It's hard.
It's hard.
Yeah, that's what I thought.
Okay, so I got another question for the computational core. It seems like, Shadowfax has grown tremendously over the years in size and scope. So how do you guys really network all of these things to work together? And is there going to be a ceiling eventually with the technology, or?
Well, we've kind of hit one ceiling already, which we're out of cooling in the data center. So in the new data center, we hope to have it so that we can actually go to probably at least 10 to 20 times our current capability. And then over time, we hope to even grow, as technology grows forward, we'll hope to grow that even higher. So on the networking side, so we're doing this as a hybrid. It's really, in some cases, it's multiple systems that are tied together through InfiniBand networks and also through the queuing system. Everything sits on top of the same file system. So that means that if you're wanting to do, sometimes we have some workflows that maybe initially you need to have large shared memory, so you bring it into the system, do some initial processing with large shared memory, and then at some point maybe you need to go into a distributed sort of computation. So having a common file system allows you guys to move your data around. Well, actually, you don't have to move your data around, you just have to kick off the various jobs at the right point to do your analysis.
And you have to have people.
People. Okay.
That's the hardest part, is finding people that know that technology. It's a subset of information technology, so you really need smart people, and when you find them, you hold on to them and throw money at them.
That's the answer to all of life's
No,
problems,
sure.
right?
Yeah.
So I got one final question for the DAC, if anyone else also would like to ask. This is a good time. It seems like we've kind of standardized into this Illumina-seq model because it's being utilized so much these days. But ion torrent technology and this mini proton also have really started taking off. So how does the DAC, I guess, prepare itself to deal with all these different data sets, or is it something you can stream into an existing pipeline?
Those are good questions. The easy answer is really I do a lot of reading, and I think Alan does too. So we do a lot of we do a lot of sort of research on our own to maintain sort of our own current state of the art. Specifically for the ion proton and ion torrent, we've already been looking at data from those. So about 2 years ago, we started looking at data from a client that has their own proton. So we're not new to that. We haven't looked at any of the like nanopore or anything like that. So I can't answer any questions like that. But really, we spend a lot of time combing the literature just to really maintain our own currency.
I'd like to ask a question of some of the future directions that Sai sees coming out of from the technology point of view, and where are the applications that you see for Virginia Tech moving forward?
So, just wanted to let you know, see, one of the common themes in all the projects that people have provided is the GRL. So we have done a lot of work for a lot of folks that have presented today, a lot of sequencing. And we are not just a sequencing facility. It's a one-stop from, from all the way from the beginning, like experimental design. If you don't design your experiment well, it's junk in and junk out. And one of the areas that we are expanding into is this epigenetics. So, David already presented some very good data. We had done the sequencing for David. We have also done projects in the plant pathology, where people are looking at the effects of herbicides on DNA methylation. So the other area that has just blossomed is the metagenomics and the microbiome projects. So, there are various labs that are looking at, what are the organisms that are living in the in the root zone, or when you treat somebody with probiotics, what are the microbiomes that show up? So, and people want to find out. There was another project that we were recently involved in is in the Patagonia and the Chile. They want to identify what are the antibiotic activity that's in there, or what are the antiviral activities in there. So those are two fields that are really blossoming, and we hope that and GRL has optimized all the protocols for all that. So we're very excited to offer those services to people at VT.
Okay, great. Well, why don't we take time to thank the panelists once again.